Translation of mRNA primarily requires tRNA, or transfer RNA, a molecule that links the mRNA codons with specific amino acids through anticodons. To ensure that the protein made is the one specified by the mRNA, the tRNAs must read mRNA codons correctly, and the tRNAs must deliver the amino acids that correspond to each mRNA codon. Now let's take a look at the tRNA structure. Each tRNA has three functions fulfilled by its structure and base sequence. The tRNA first binds to the mRNA. The tRNA is actually made up of a polynucleotide chain. The tRNA binds non-covalently to the mRNA with hydrogen bonds, and the anticodon located at the proximate midpoint of the tRNA is complementary to the mRNA codon that the tRNA reads. As follows, the tRNA binds to the particular amino acid that corresponds to the anticodon. This binding is covalent and very specific. The tRNA binds to a specific enzyme that attaches it to only one out of the 20 amino acids. When tRNA is carrying an amino acid, the tRNA is said to be charged. Aminoacyl tRNA synthetases is the family of enzymes that charges the tRNA with its correct amino acid. First, an amino acid and ATP attach to the synthetase. Then, two phosphates are removed to form AMP. The tRNA then joins the synthetase. Then the AMP and charged tRNA are released. This reaction forms a high-energy bond between amino acid and tRNA, and the energy in this bond is used later in formation of peptide bonds. Each enzyme is specific for one amino acid and its corresponding tRNA. That's a lot of enzymes. It's also important to mention the Wobble phenomenon, or the way a cell gets by with 40 tRNA species with anticodons for 61 different codons. Because the specificity for the base at the 3' end of the codon and the 5' end of the anticodon is not always strictly observed, in some cases unusual or modified nucleotide bases occur in the 5' position of the anticodon. For example, the amino acid inosine can pair with A, C, and U, so that anticodon 3' CGI 5' can bind with three different alanine codons, GCA, GCC, and GCU. We must also note that Wobble occurs in some matches but not others, and more importantly, it does not allow the genetic code to be ambiguous. In other words, each mRNA codon still binds to just one tRNA species carrying a specific amino acid. The slight changes in anticodon sequence due to Wobble simply allow less anticodons to work for more codons. Lastly, charged tRNAs interact with a ribosome in the cell. The ribosome has several surface sites that specifically fit the 3D structure of a tRNA molecule, and they bind non-covalently. We'll talk more about specific ribosome structure in the next video.